So the first step of the gram stain procedure is to get the bacteria onto a microscope slide. So you can see I have a microscope slide here and a wax pencil. I'm just going to draw a circle on the wax pencil so that I know where the bacteria is. This particular sample is Micrococcus, so I'm going to label it with an M so that I know which slide this is. Next, I'm going to turn on the Bunsen burner and I'm going to get the bacteria out of the tube using aseptic technique. So as before, we sterilize the inoculation loop, grab the cap of the test tube, flame the opening of the test tube, get some culture on the end of the inoculation loop, and deliver it to, in this case, my slide. I'm just going to grab one more loop full of bacteria, maybe two more loopfuls, just to make sure I have enough. You can also get bacteria off a petri dish from a colony, and in that case you probably don't want too much. You want a decent smear but not too thick. So the last step that I need to do is heat fix the bacteria to the slide, and that basically means dry them and have them attached to the slide so that it doesn't wash off during the gram stain procedure. So what I want to do is put this through the flame, not too much that I'm cooking or frying the bacteria. I don't want to denature all the proteins, but what I want to do is put enough heat on there. So the way I do that is I pass it through the flame, something like this, about 3 or 4 times. Now I'm going to let the microscope slide dry before proceeding to the next step. We're now ready to do the staining part of the gram stain. So there's my microscope slide, and the first step I'm going to add the crystal violet. So what I do is I add the crystal violet to the slide, put it on top of the bacteria, and let it sit there for 1 minute. At the end of 1 minute, I'm going to wash off the excess stain, and what I'm doing is I notice I'm flipping the slide upside down. I don't want to have the water hit the bacteria directly because that will blast the bacteria off the surface of the microscope slide. So what I'm doing is I'm putting the water on the other side, and it's going to gently curl around the glass and wash away the excess stain very gently. So once I'm done that, I can dry it off on a piece of paper towel by just tapping it, and I'm ready for the next step. Step number 2 is adding the grams iodine. Similar to the crystal violet, I'm going to add the iodine onto the bacteria and let it sit for 1 minute. At the end of 1 minute, I can wash off the excess iodine in the same fashion that I used to wash off the excess crystal violet. So the third step is the ethanol wash. So here's the ethanol. So with the ethanol wash, I have to be careful that I don't let it soak for too long, because that will dissolve all the crystal violet. I just want to have it on there long enough to get the crystal violet away from the gram negatives, but not dissolve it from the gram positives. So I'm going to add the ethanol on there. I don't want to leave it much longer than about maybe 20 seconds. At the end of 20 seconds, I'm going to let the ethanol come off, give it a quick rinse, take a look at the slide. If there's too much purple on there, and I think our slide is actually looking pretty good, if there were too much purple on there, I would do another ethanol wash step. The last step, I'm going to add the Safranin or Safranin red. So very similar like the other steps, you put the stain onto the bacteria. In this case, I'm not going to leave it quite as long as 1 minute, maybe 30 seconds to 45 seconds. At the end of that, I'm going to wash it one more time. So now I'm going to let this microscope slide dry, and then we'll look at it under the microscope.